As ever, we have two excellent speakers for you. So without further ado, the first speaker is Federico Baltar, who's going to talk to us about pelagic fungi and their role in carbon cycling in the open ocean. So Federico, over to you. Thank you.
Okay, thank you. I'm gonna share the screen. Hopefully should be fine. Let me know. Can you see my screen?
Yeah, that's coming across great.
Okay, perfect. So then I will start. So should I start now or should we wait some more minutes? Whatever you want. Should I start now?
Sorry, I realized I was on mute. Yes, please do start, and I'm sure people will come in as they.
Okay, perfect. Good. So hello everybody, and thanks for inviting me to present here. I'm happy to have the chance to share the little we know about the fungi, in this nice forum. Well, this presentation, as Adrian was saying, is going to be talking about the role of fungi, in this case, mostly focusing on pelagic fungi, in the water column, in the carbon cycle. And not only carbon cycle, but we are going to also be talking about nitrogen cycle in general, also. Good. So, and I should mention, this is also work that has been done in collaboration with other people, also from the University of Vienna. Let's see how they have or Eva Breyer. So, I should know, when we talk about microbes, probably all of you in this forum know, microbes are very important because they are the main engines driving the elemental cycles. Why is that? Well, not only because, of course, they have the enzymes involved in many of these important transformations, and they are the main producers and the main consumers. So the main producers, as you very well know, they're fixing the phytoplankton, fixing CO2, and then being grazed by zooplankton, ultimately affecting how much fish you're going to get on the table. But also because they're going to be controlling, they're not only the main producers, but also the main recyclers. So you know that a big part of the primary production, or usually around half or so, is going to end up as dissolved organic matter through different processes. And then this dissolved organic matter is going to be consumed by heterotrophic prokaryotes, and then by consuming this and incorporated into the biomass, and then being grazed by small grazers, they reintroduce this carbon back into the classical food chain through what is called the microbial loop. So this microbial loop, as you can see in this diagram, it's placed in a very central cornerstone position. At the end, here, the amount of carbon that is going to be sinking down and is going to be sequestered, it's going to be affected through by the microbial loop, but also the fate of how much of the carbon is going to end up in the fisheries, or how much of the carbon is going to end up respire and going back as CO2 into the atmosphere. So ultimately, this microbial loop is very important. But this has traditionally been focused or been developed mostly thinking on prokaryotes. Originally, it was bacteria mostly, towards the end of the 70s, when end of the 70s, beginning of the 80s, when the microbial loop concept was generated. And the question is, and then lately, in the last decade, a bit archaea also known to be contributing. The question is, what about fungi? Well, fungi were not even supposed to be in the open ocean. So recently, like in the last 5-7 years, 5-10 years, not even 10, less, fungi has been starting to be showing up, in molecular studies. And also in other ecological studies, now we know there is something called the microloop, for example. The microloop is when some fungi, some parasitic fungi, usually chytrids, they are able to parasite on diatoms, and then by doing this, they also release zoospores that can be consumed directly by small zooplankton, and by doing this, they make a loop in the carbon cycle, what is called the microloop. Okay, that's something that has been developed in the last few years, but we know that in the ocean it's not only parasitic fungi, there are also saprotroph. So what about this? No? Well, this we're going to be talking quite a bit today. So I don't have, well, I'm going to try in the next 15 minutes or so to summarize the findings that we have that we did in these 3 different papers. So I put it so you have more time, you can dip into it, okay? But basically, I'm going to give you a bit of the appetizer and put everything a bit together so that you can, then if you're interested, you can go further. So in this first paper, we look into the global, the global ocean role of fungi utilizing proteins. Then the second one was looking at the carbohydrate utilization. And the third one was one in which we did the same, well, looking at the carbohydrate and protein utilization, but in this case, looking at prokaryotes and not so much on fungi. So now a bit into more into data. So all these papers, or well, not all, but most of it was generated from data from the Tara Oceans. No? The Tara Ocean is this global expedition, probably most of you are aware, no? That went all over the place, or well, around covering a wide area, sampling scheme, no? Different regions. And they were collecting data for not only diversity, but also for metagenomic and metatranscriptomic. No? When I talk genomic, I talk about gene content. When I talk about transcriptomic, I talk about RNA, I talk about expression. So the first thing we wanted to do was first to see if there were caseins that were affiliated to fungi. Caseins are the carbohydrate active enzymes. So these are the enzymes involved in the degradation of carbohydrates. And then we looked for it, and we were happy to find that they were there. So that's good. So there are fungi that has the potential, but are they doing this? Well, that's a different question. So for that, we need to look into the expression. And when we look into the transcription, we find that there were transcripts of fungal caseins. So we were very happy because we could say, okay, fungi are actively involved in the degradation of carbohydrate in the ocean. And then in this diagram, well, in this plot, we see that there is also like this cluster by depth, which is also it's not just something random. This ecologically makes sense because the same happens for prokaryotes, for example. So you see the mesopelagic here is on the red, on one side. It's not important the circle or the triangle for what I want to say, just different size fractions. It doesn't matter. And the epipelagic is more on the left, no? And not only for the gene content, but also for the expression. So this was good. We could say, okay, fungi are actively doing something here. It's not only bacteria, no? Or here. And it makes ecological sense. Also, something interesting is that we found that there was a tight link between the genomic potential, so the metagenomic, and the expression, the transcriptome. And this actually we found not only for the caseins, but also in the following study when we look into the proteases. Okay? So this basically says, what does this mean? This means that when there's a tight link, it means that what is there is quite active. This is what it means. Also, something that that was also interesting for us, because we had these 2 different studies, no? One looking at proteases, no, degradation of proteins, and other caseins, degradation of carbohydrates. And when we actually put it together, we realized that there was, as you can see here, no, this is caseins. This is the metagenomic in red, transcriptomic in bluish. We see that there is a tight link also. This is strongly related. The fungal protease is the abundant and expression, no? Both looking at the metagenome and the transcriptome, is strongly related to the fungal casein, which makes sense also. So this also makes it was also good for us because it's like, okay, well, there's not much carbon, so whenever there is a degradation of carbohydrate by fungi, there can also be degradation of proteins, no, by fungi. So this was also good for us to find. And also, of course, also the fact that we found also transcripts of fungal proteases, which also involve fungi, then not only in the carbon cycle, but also in the nitrogen cycle, no? Because the proteins also have nitrogen. Okay, here I'm going to show you basically this is a summary, no? Because of course I don't have time to show you all the individual plots for all of this, but here you have like the depth pattern, right, for fungi on the left and for prokaryotes on the right. I don't know if you can see my mouse. Can you see my mouse? Oh, yeah, also good. So here you have, so fungi, prokaryotes. And here, what we have is in blue is for the caseins, and in green is for the proteases. So carbohydrate and protein degradation, no? Utilization. So, and here you have for the abundance of the casein or the proteases, diversity of caseins, proteases, expression, or the percentage of secretory also, what is predicted, how much they secrete. No? And here, what we found was that for fungi, interestingly, when going from the epipelagic to the mesopelagic, everything seems to be relatively increasing. Whereas for prokaryotes, this was not the case. No? For prokaryotes, there are some things that were increasing, others that were not. No? For example, we found that not the total proteases, the total caseins of prokaryotes were not really increasing with depth, which is in contrast with the fungi. Not everything increases with depth. You know? So why were we interested? Because at the end, we don't only want to study how fungi are doing, what is their role, how does this change with it. We also want to put it into context of what we know more. So how does this compare to what prokaryotes are doing? Prokaryotes are also heterotroph. So the question is, are they doing the same thing? Are they competing with each other, or do you have the same ecological niche or not? No? This is why we thought that to try to put all this together could be informative. So, for this shows that that it starts to give the first glimpses of showing that it's not the same for everybody. No, that at least the changes with depth, there is a different contribution or a different role, at least with depth, when it comes to the degradation of casein and proteases between prokaryotes and fungi. Okay, good. So, but also here, when you look into the tail, this basically what is increasing with depth is mostly the percentage of secretory. No? This is very interesting because the percentage of secretory, which also increases with depth for fungi, and this happens for casein and proteases, seems to be a kind of a conserved feature, because this also happens for prokaryotes. This we knew from before. And this highlights the importance of secreted enzymes in the degradation of organic matter, particularly its relative importance more towards the deep ocean, which has been traditionally used in the context of marine bacteria as indicative of a preferential particle attached lifestyle. So maybe also this is showing that fungi are also preferentially particle attached in the deep. But this is also the first glimpses. No? But as I will show you now, the composition of the enzymes that they actually secrete can be different, no? And pointing to maybe different strategies. So what did we do also? What we were able to look into the taxonomic affiliation. Taxonomic affiliation just basically is giving a name, an owner for the genes or the transcript that we find. No? So here I have an example. So here you have, this is for the caseins, for example, and here you have here on the top, these are the Tara regions, no? The Indian Ocean, the Mediterranean Sea, the North Atlantic Ocean, North Pacific, the South Atlantic, Southern Ocean, South Pacific Ocean. Okay? And here you have the taxonomic affiliation, and you have which fungi were the owners of these casein genes. No? And here you see mostly throughout the ocean is dominated by the Ascomycota, except maybe in the Mediterranean Sea, which is dominated by the Acidimycota. No? Of course, this is a quite general taxonomic level. We can also go deeper, and we actually did, to show specific classes, no? Not only at the phylum level. But also what we can do, or we could do, just to give an example, is not only to look at who is the owner of these genes, but also what is the diversity of the functions of these genes. No? For example, here, which casein functions, casein classes are they using? So again, here by the different regions, and here, for example, you see that this basically the glycoside hydrolases or the glycosyl transferases are mostly dominated, no? Throughout the whole ocean. No? Good. So now what I'm going to show you, these were just examples of what we could do, and here is going to be the summary of the taxonomic affiliation, which is going to be here in the left always of each, and the functional diversity, which is on the right of each of this square. This is summarized. This there's a lot of information summarized there. This is in a 3 paper I also published recently. So if you want to, well, I will mention about this later anyway. But anyway, so here you have, and this is comparing for the fungi and for the prokaryotes. Fungi for prokaryotes for the utilization of carbohydrates and proteins. And here we have the this is for the epipelagic and this is for the mesopelagic. And we actually distinguish between the total and the secreted fraction. Okay, so briefly, what did we find? Well, for us, we were first happy to know which are the fungi because we didn't know who was doing this. And we found that the carbohydrate utilization, by the way, I should mention, all this is based on transcripts. So this is not potential. This is based on the actual expression of the transcription of genes. So the main fungi performing these activities for the degradation of carbohydrates were the Dotiodiomycetes and the Malassesiomycetes, okay? Which also happened to be the same one that were doing this for the proteins. So are the same fungi doing both things. So that's good. And then, interestingly, as what we found also was that when you look into the mesopelagic, there is a shift. So the fungi that are actually performing the carbohydrate utilization or harboring or being the owners of the cazymes, they change to the Letiomycetes and the Erotiomycetes. Interestingly, the same happened for the proteins. So also there's this shift in the taxonomic affiliation, which fungi is doing this. But they are the same fungi degrading the carbohydrates, the protein. When it comes to the functional diversity, there is not big changes in the cazymes, in the carbohydrate utilization. It's basically dominated by glycoside transferases, glycoside hydrolysis, and auxiliary activities. And this doesn't change with depth. It doesn't. It's quite conserved, and also between the total or the secreted fraction. Okay, so how does this compare now to the prokaryotes? Well, to the prokaryotes, what we find is that the main bacteria doing the carbohydrate utilization are the alpha, so alpha proteobacteria, followed by bacteroidetes and the gamma proteobacteria. But when we move into the secreted fraction, this also we would expect the gamma proteobacteria are the one dominating because they are the ones that secrete the most. And also the bacteroidetes become less important when you go in the mesopelagic. When we look into the prokaryotes in the protein utilization, the same happens. It's dominated by alpha proteobacteria, but they're secreted by the gamma, and then the bacteroidetes also becoming less important in the mesopelagic. When we look into the functions, here, we look into the functional diversity for fungi, we find also very conserved as we observed for the carbohydrate. So we had here, in this case, dominated by serine peptidases, metallopeptidases, cysteine peptidases throughout the water column and the regions. When we look into the prokaryotes, this a bit shifted. So you have the metallopeptidases more important than serine peptidases, and also quite constant, although you have these changes between the secreted and the total. No? Like, for example, you have the peptidase inhibitors. Anyway, this is just to give you, well, this was just going one by one, but I just actually did it together because we're not in a room, so we are online, so we have the screen, so we can see the whole size. Good. So if we put all this together, this is basically a summary. So what can we know? What can we get from here? Well, is there a taxonomic affiliation shift with depth? Yes, for fungi and for prokaryotes. Okay, good. We didn't know. Is there a functional diversity shift with depth? Well, for prokaryotes, yes. For fungi, no. Is there a functional diversity shift between different fractions, the total and the secreted? Prokaryotes, yes. Fungi, no. Is there secretion of cysteine peptidases? Fungi, yes. Prokaryotes, no. Secretion of glycosyl transferases? Fungi, yes. Prokaryotes, no. So at the end, what this is also is showing is that there seems to be a contrasting strategy. So there's some things in common, but also different between what the fungi and the prokaryotes might be doing in relation to degradation of proteins and carbohydrates. Okay, so now to basically put it together, and then I have another slide at the end, so I'm not finishing yet, but, but just to put it together. Well, now we know that fungi are everywhere. So we are also looking with many other things, no, looking into diversity and biomass of fungi in the global ocean. We find them almost everywhere we look. And now we can also say they're active contributors. So it's not just, they're not just passive spores floating around, because if not, they wouldn't be transcribing, you know, all the genes. So they're active contributors to the carbon and the nitrogen cycle. Also, we see that there are tight link, as I mentioned before, between the potential expression, showing that maybe there's not a lot of fungi, but what is there seems to be active. Also, there's a close coupling between the degradation of proteins and carbohydrates, which makes sense, which also means that whenever there's degradation of carbohydrate by fungi, there's also degradation of proteins, whatever they are, probably attached to particles. Also, we have seen evidences now that they are suggesting contrasting strategies for the enzymatic degradation of organic matter between prokaryotes and fungi. So maybe this suggests that fungi and prokaryotes might occupy different ecological niches. We might share some, but might be also a bit different. So, and this was also part of the main aim of this presentation, also of this paper, was actually to say, okay, well, if fungi are there, if fungi are active, then we should also include the saprotrophic fungi as active members and important members of the microbial loop in the ocean. So if you want to read more, this is what we mentioned here. This was the main aim of this paper, I think. The main message I wanted to come across. This was just published a few months ago. And I recommend you to, if you have time, if you're very bored or you're curious, to have a look into it. With this, I would just like to thank you for attention, all the people in my group, and also to acknowledge two projects from Jetson where I'm collaborating at the moment, the Impact in Spain and the Tower from China. And thank you very much. If you have any questions, I'm happy to answer.
Excellent, Federico. Fascinating stuff. I have to admit, we don't hear a great deal about fungi. I think so it's fascinating to get this insight in terms of, you know, what I guess may be a neglected part of the ecosystem. So let me just check on the chat. It's a bit difficult. So if people want to add questions to the chat, that's fine. If they want to just put their hand up and ask a question, that's fine. We've got a good 5 minutes or so. So I can use my position as the person organizing the call to ask the first question. So, I mean, it's fascinating to me. Do you have a feel for, you mentioned that the prokaryotes and the fungi potentially have this sort of different ecological niche. Do you have a feel for what the fungi are preferentially feeding on? Is it relatively fresh material that's sinking down? Is it the more refractory material? Is it within fecal pellets? Is there any way of telling that?
Yeah, this is a very good question, because well, first, also answering to your whole comment from the beginning, the reason why you haven't heard much about the fungi in the ocean is because we don't know much. So this is the reason, and most of this, as you can see, is relatively new. No, it's in the last couple of years, really. So that's the reason. So this means that what I can tell you at the moment is more my feeling, more than 100% sure answer, okay? So I think we have to distinguish, no? Because as I showed you before, so we have different types of fungi. So we have the parasites that are going to be more associated to diatoms, and of course they're going to be getting fresh material directly from the diatom. No? And then you have the saprotroph. And then the saprotroph is a bit like what happens with the bacteria. No? You can say, what kind of organic matter bacteria are using? Well, it depends, right? If you're in the surface, you have usually the bacterial community is more used to use labile material. Where you are in the deep, they are more adapted in general. If they are in the water column, to be using refractory material, not if they are attached to particle in the deep. Then, of course, they are more used to get the fresh material. So I think a similar thing would be safe to say for fungi. I think you will have fungi besides the parasites, of course, you have the saprotroph. You're gonna be using. There will be some of them that can use. They will be used to use larval material, but I can also envision that probably there are gonna be some fungi that are able to degrade refractory, even more refractory material maybe than bacteria or than most of the bacteria, because we know this happens also in soils. So maybe there's something like this. But this is just a bit of what I think is at the moment. So let's see. Maybe in some years I can tell you something more precise.
Yeah, I mean, it's just fascinating to hear you sort of speculate in terms of how it might be. And so do you think that might be partly behind the shift in the community structure that you see with depth in terms of going into the mesopelagic?
Exactly. That's exactly it. Exactly it. This is exactly because this will happen also with the bacteria, right? So bacteria also you have this shift usually between the main dominance, no, in the surface, not usually dominated by the alpha proteobacteria. No, you have usually dominated by the SAR11, that is more oligotroph, no, or you have when you have more rich conditions, you have the Roshobacter, no, which is also alpha proteobacteria, and then you get into the deep, and this changes completely to other organisms. For example, you have more suddenly the Thaumarchaeota, and also you have the other, the SAR324, for example, or the SAR202, which are more adapted to deep ocean organic matter conditions. So I think it would be safe to expect something like that also for fungi.
Fascinating. So we've got a couple of questions in the chat. Nathan, do you want to ask your question?
Can you hear me?
Yes, we can hear you.
Yes, you can.
Hi, Federico. Thank you for the talk. I'm wondering what is the proportion of fungi VS compared to the prokaryotes in terms of cell abundance in the water column?
Yeah.
Or numerous are they?
Yeah. Thanks, Nathan. That's also a good question, and thanks for your nice comment. Yeah, this at the moment actually we are finishing a manuscript now on this. So we have been collecting numbers and biomass of fungi. We're using 3 different methods in parallel from different oceanic regions. And I don't have the actual numbers now, like I don't remember from the top of my head. But yeah, of course, I can tell you when you talk about prokaryotes, we have to distinguish between bacteria and archaea, right? Because bacteria is also much more abundant than archaea, well, depending where you are, but in general. So prokaryotes are more abundant than fungi. This I can tell you. Yeah. But I would say if you tell me from, like, to give you a number now, because I don't remember, I would say it really changes between depth and between location, but what can I say? Maybe around it really changes a lot actually. So I cannot tell you the average, but let's say I'm gonna tell you to be safe, there are less than bacteria and less than archaea, but no like 1 order of magnitude less than archaea, I would say.
Excellent. Thank you.
Thank you.
But if I'm not sure whether this is a quick answer, but just in case it is, Clarissa, do you want to ask your question?
Sure. I was wondering about the morphology of the fungi. Do you know how large they are and like if they have any unusual shape, or are they just like bacterial cells?
Yeah, no, this is also a good question. Yeah. Now we know because of this paper that we're finishing here. We one of the methods, actually no, 2 of the methods are based on microscopy. No, we're done with fluorescence in situ hybridization and with Holcofluor. Because of this, we have to individually go cell by cell and measure the biomass of each of the cells. So now we have a good idea of how they look. Interestingly enough, most of the fungi we found are yeasts. So they're yeast. So you find filamentous more maybe associated to particle, but if not, when it's in the water, and the small particle is mostly yeast. So they are more like unicellular. And the size, it ranges, but I would say with a big confidence that most of the ones we find are between few micrometers, between 2, most of them, 2 to 8 micrometers.
Thank you. That's fascinating.
Excellent.
Yeah. Welcome. We actually have been also collecting we have a collection now of fungal cultures, like 300 also, or that we have managed to get from the cruises in the last 5 years. And also there we have, I would say, in that case we have, it's also dominated by yeast, but also we have hi-fi fungi, maybe 20% or so.
Excellent. Thank you very much. So I think we need to move on now. But thank you once again, Federico. Absolutely fascinating. And so if people have any questions, I'm sure he'd be happy to answer them. So I think he's got to head off now, so I wouldn't so much put them in the chat as maybe email them in. But thank you, Federico.
Thank you, Adrian. Thank you, everybody, for paying attention and asking the question. Thank you. Bye.
So if you could stop sharing, then that would be great.